So now we're going to start part 4 of our chapter 9 lecture. And we've fully oxidized glucose through the Krebs cycle, or the citric acid cycle. And we've ended up with a total of 4 ATP. So where's all the ATP? We need to produce quite a bit more in order for our cell to live and do all that it needs to do. So we're going to look and see if we can't find it in the next and last step of cellular respiration, which is the oxidative phosphorylation phase. So this coincides with section 9.4 in your textbook. And basically what this is going to be looking at is that during oxidative phosphorylation, we have chemiosmosis coupling the electron transport to the synthesis of ATP. So we're going to look and review and remember what all these terms mean. But basically, following glycolysis and the citric acid cycle, we have produced and harnessed energy in the form of electrons that are stored in the NADH and the FADH2. And that's where most of the energy that was extracted through all those redox reactions as we went through the catabolism of the different carbon compounds. So these two electron carriers are going to donate their electrons to the electron transport chain, which is going to power the ATP synthesis through oxidative phosphorylation, similar to the process that we studied with photosynthesis. Oxidative phosphorylation accounts for 90% of the ATP that is generated by cellular respiration, which is good to hear since we've only gotten a total of 4 ATPs up to this point. The ATPs that we have been able to get from the glycolysis and from the citric acid cycle have all been formed through what we call substrate level phosphorylation. And what that means is that the phosphorylation of ADP is actually occurring through the breakdown of a molecule. So the phosphate, inorganic phosphate, is being added to the ADP by being taken off of another organic molecule. And that is very different from the type of ATP formation or synthesis that's going to occur in the electron transport chain. So I want to make sure you understand that there's a difference between the process that we get ATP up until this point versus what's going to happen through oxidative phosphorylation. For each molecule of glucose that's degraded to CO2 in substrate level phosphorylation, we actually have the use of an enzyme. So this is all that this is showing. The electron transport chain is located in the inner membrane of the mitochondrion. So it's within the cristae of the mitochondrion. So we have all of our citric acid cycle occurring within the matrix, and the NADHs and the FADH2s are now carrying electrons, and they need to end up in the membrane of that mitochondrion. That electron transport chain is made up of proteins that are located within the membrane. And again, very similar to the electron transport chain in photosynthesis, we have a series of reduction and oxidation reactions occurring that are going to pass the electrons on in order to generate the energy that's needed to create a chemiosmosis. Actually, what we really want is a proton gradient, similar to what we find with photosynthesis. This is the final portion of our diagram showing where we're at in the process of cellular respiration. So we're at this last stage. And I just want to talk a really short amount of time about the NAD plus molecule or NADH molecule. So basically, NAD plus is a coenzyme and also an electron acceptor. NAD plus functions as an oxidizing agent, and every NADH molecule, which is the reduced form of NAD plus, represents stored energy, which will be used in our oxidative phosphorylation process. This is the structure of an NAD plus molecule. You don't need to memorize this. I just want you to be familiar with it and have a chance to see it. And this is just showing that the process of reducing NAD to NADH. NADH is going to pass its electrons to the electron transport chain. And unlike an uncontrolled reaction, the ETC passes its electrons in a series of small steps instead of one explosive reaction. If oxygen were not present, we wouldn't have a final electron acceptor at the end of the electron transport chain. And so the presence of oxygen is needed. Again, we needed it for pyruvate even to enter the citric acid cycle, because this all is going to be, actually you can think of oxygen as the magnet that's pulling this reaction along, starting with the citric acid cycle down through the electron transport chain, with the oxygen waiting there to be the final acceptor. If we were to just take some hydrogens or hydrogen gas and combine it with oxygen to end up with water, we would actually end up with, there is such a huge drop or change in free energy. We would actually have an explosive reaction if it was just one straight reaction. So that's why, and the beauty of the electron transport chain is by having these small reactions in between, we're able to actually control the reaction and actually gain the energy and harvest it in a way that it's not just all lost as heat energy. Another way to think of the electron transport chain is to think of the electrons as flowing downhill. So we saw that a little bit with photosynthesis, how there's a stepping down process, and every time the electrons get passed down, there's a little bit less energy available in the new molecule that's being synthesized or formed. So going from carrier to carrier down through this process, each carrier is becoming more electronegative, and that again is going to help draw these electrons down the process. And then again, the final destination is for these electrons to combine with oxygen and allow oxygen then to combine with hydrogen ions to form water. This is showing how we have this change in free energy starting from the first reaction. So we have our NADH here donating its electrons in the first reaction at the highest free energy level. And then as we move down, step by step by step by step, we're also moving down in free energy until finally we're able to donate the electrons here to the oxygen, who's again very electronegative, ready to take those on. And then there's an immediate synthesis of water. One thing to note here is that FADH2 actually joins this electron transport chain a little bit further down than the NADH. So you can see, I mean, it maybe isn't even very easy to tell from this diagram, but there's a real reason for that, and it's because NADH, the electrons in this molecule actually have more energy than the FADH2 molecule. So there is a difference in the starting free energy of these two electron carriers. So that's why one will start up at this first reaction, and then the other actually will join the electron transport chain a little bit further down. The big blue blobs there are what are the multi-protein complexes that are located within the membrane of the mitochondrion. And these are just some facts from your textbook about this electron transport chain. Cytochromes are some of the proteins, the names of the proteins that are within that. And actually, we call those cytochrome complexes. And we saw one of those in the photosynthesis electron transport chain, had one of those complexes. So these are, again, it's another name that is used for the proteins that are located in this, the inner membrane of the mitochondrion. It's important to also note here, one important fact I see is that the electron transport chain does not generate ATP itself. So it's actually similar to photosynthesis, and we'll see why that is in just a second. It has to do with the fact that our ATP is actually going to be synthesized through chemiosmosis, just like it is in photosynthesis. So as electrons are transported down the chain, proteins pump hydrogen ions from the mitochondrial matrix into that intermembrane space, which remember is the space in between the outer membrane of the mitochondrion and that inner cristae membrane of the mitochondrion. So in that fluid-filled space, we get a buildup of hydrogen protons. Then the hydrogen protons move back across the membrane, going through that molecule that we learned about before called ATP synthase. And ATP synthase uses that flow of hydrogen protons to actually drive the phosphorylation of ATP. And that is the definition here of chemiosmosis occurring in cellular respiration. This is a picture of that ATP synthase, and you can think of it as a little motor, again, that is turning as the protons pass through. And it's actually the turning process that generates the energy that's needed for this phosphorylation to occur. This is another picture of our electron transport chain, and you can see how they're embedded. These complexes are embedded in the membrane, as well as the ATP synthase. And so oxidative phosphorylation involves these two processes joined together. We need both the electron transport chain in order to get our hydrogen protons into the intermembrane space here, so that then we get a gradient that's going to drive those protons to a lower concentration area outside, generating our chemiosmosis to make ADP into ATP. So chemiosmosis happens because we've set up a hydrogen proton gradient, and the ATP synthase is allowing the hydrogen protons to flow. And that power is bonding in